that introduction. And also thank you everyone for coming. I know this isn't your usual time. It is 7 a.m. my time. So yeah, I am very excited to talk to you, of course. I'm here to talk to you about matters of scale in ecology and evolution. So what I've done is I've actually recast some of my projects to kind of give us some general lessons about different aspects of scale. And so what I want to do is start off with this quote about community ecology. In about the year 2000, say circa 2000, reports of my death have been greatly exaggerated. And I love speaking to more people with evolutionary backgrounds that kind of give you an update of how far community ecology has come in the last decade or so. But back at this time, people made this argument that communities were basically too complex, that they could never achieve the fine scale mechanistic power of population ecology, or really this broad generality that you can get from macroecology. So it's kind of like stuck in between this problem of not being mechanistic enough and also not being general enough and being too complex overall. But of course, populations are embedded within communities, and communities are embedded within geographic landscapes. And so really the community is this really key important characteristic scale in order to understand what's happening at these other levels. So you can't really study populations without knowing what's happening in their communities, or at broader scales, what's underlying some of those changes in biogeographic patterns. And so really in the last bunch of years, community ecology has really been focusing on addressing these issues and becoming more mechanistic, becoming more general. And I'll tell you one of the ways that I've been addressing this. So I do work primarily with a study system, annual plant communities from Mediterranean climates, although we are moving more into other systems like duckweed and beetles right now, which is fun. But these annual plants that I work with are great for a number of reasons. First, the communities that I work with are hyper diverse. They tend to fall in biodiversity hotspots. Annual plants are extremely amenable to experiments because you can manipulate things and move things and actually track all fitness components within a single growing season, which is great. But the specific way that I like to address the mechanisms that underlie community patterns is to actually think about communities as sort of conglomerations of populations that are embedded within them. So you can think about these three species here that are each doing something different. Here it's an environmental gradient. They're all kind of sorting differently along this environmental gradient. But basically what happens when they're all put together and they have these different environmental associations, but they're all interacting with each other. So it's very much a multi-species population ecology type approach is what I would describe my approach here. So yes, populations explaining community patterns. And what I've been doing in my lab is first trying to build up the community ecology side of things. Can we predict when and where and why species can coexist, how that varies through time, how individual population persistence underlies that coexistence, and then take those lessons and apply them to better understand evolutionary ecology. And so there's this very strong feedback in my lab between better resolving the ecology and the mechanisms that promote or hurt coexistence and persistence, and then trying to understand that feedback loop with evolution. And so what I'm going to do today is this talk is mostly broken into two parts. I will say this is more on the ecological side of things with a bit of evolution, but there is an evolutionary ecology component to this, but I'm not going to go as hard into it as some of the work we've been doing the last couple of years, but I will talk about it. So I'm going to start with the community ecology side of things for part one. And so what I want to start with is this idea of the filtering metaphor. It's something that a lot of us are familiar with. It's something that we teach our undergraduates often as part of their core ecological courses. And just as a brief refresher of what this is, it's the idea is that there's some regional pool of species that are kind of like floating around, and those species have a whole bunch of different phenotypes. They're very diverse. And the idea is, how do we get from that regional species pool down into that local community? And people have thought for the last series of decades that there are these series of environmental filters. The first being kind of only letting species through that match the abiotic environment of a particular area. And then a second filter, which basically then filters out of those species that can live in a particular environment, how many of them are well matched to that local community. If you saw a pattern like this, where local communities tended to be composed of species that are really similar in some ways in their match to the environment, but also really dissimilar in other ways, indicating that they might be able to compete less strongly with each other, this is what we would think of as being evidence of strong filters shaping that community. But of course, environmental filtering is not likely independent of competition. And we actually don't have a lot of empirical examples that really demonstrate this super well. And the idea here, this is a conceptual schematic from a paper from Functional Ecology from 2015. It's basically the idea that environmental filtering in a sensu stricto sense is the idea that you have a species that can live in one environment. Here it's this little gray environment. And if it were to disperse into this adjacent habitat with a different environmental condition, that it would be unable to persist there. And the idea is that it's been filtered out of that white environment and it's filtered into this gray environment. But of course, you can get a similar pattern due to competitive exclusion. So here's a species here, this yellow species that can live in both environments. So it wouldn't be filtered out from the white environment if there were no other species there. But in this case, if it's actually forced to contend with this one species that's a better competitor in this white environment, that's where you actually see its absence from that environment. And so what you can see is that basically the outcome you would expect here is indistinguishable from environmental filtering, even though it's driven by environmentally based competitive exclusion. And what's interesting is that, you know, as I've said, populations are embedded in communities. And so it really doesn't make sense to think about environmental filtering without the perspective of other competitors in the community. Despite this, about 85% of studies actually confound competition with environmental filtering when we're trying to actually assess the strength of this environmental filtering. So I have two questions here. Is evidence of environmental filtering consistent in the presence or absence of competitors? And does the answer here differ if we contrast among different abiotic environments? So if we're comparing a dry environment versus a wet environment, how does that affect our conclusions about filtering? And what we would want to test this is, again, here's our metaphor here. So again, we're seeing that there's these three species that have passed through those two filters and they can succeed there. What we would want to do is actually do an experimental manipulation where we take species that had presumably been filtered out by those environmental filters and actually force them to be in that environment. And if in fact filtering is responsible for shaping those communities, what we would expect to find is that they would be unable to persist there. And so to test this, I'm not going to go too in detail into the methods, but how do we measure this persistence? The way that I've been doing this is using invisibility experiments. So basically in a greenhouse, what I would do is set up an experiment like this, where we have some focal species, this little aster species here, and I either grow it alone in the absence of competitors in a particular abiotic condition, or I grow it with competitors. And not just as simply as this, there's actually a whole model fitting thing that underlies this, but this is kind of the idea of what's happening here. And through this, I can actually fit some population estimates of how those focal individuals do when they're trying to invade this competitor-free environment, which I'm calling the invasion of just E, which is the environment, versus in the presence of that competitor. So it allows us to say, you know, basically how are these focal individuals experiencing the environment in the presence and absence of competitors? If these values fall lower than 1, this is a case where you would expect this is a population that is not going to be persistent. Whereas if it falls above 1, it's a population that you would predict is going to be persistent. We replicate this in 2 soil environments, a wet and a dry environment, just to see the sensitivity of competitors to these different environments. Okay, so the first thing I want to show you, conclusion here, is that environmental filtering only emerges with competition. And so what I have here on the x-axis are the 2 environments. We have a dry soil moisture environment and a wet soil moisture environment. And again, we're looking at that invasion growth rate, specifically in the absence of competitors here. And this line is that persistence threshold. So above this line, they're predicted to persist, and below it, they're not. So if I just slap the data on here, basically what you can see is that almost all of these species, regardless of whether or not it's a wet or dry environment, are actually predicted to persist in that environment. So we're thinking the percentage of species that are filtered out and are unable to persist there, basically none, just like this one, which might have even been an anomaly, who knows. If we compare this to what happens with competitors, exact same plot, but now with competitors, you can see that many of these species pairs are actually driven below that persistence threshold. And in fact, if we were to break this down to the number of species that are actually filtered out, you can see that it's quite a lot of them. And also it's pretty equal among these two environments, which is kind of interesting. What I've done here is I've just highlighted a particular species pair. So you can kind of see that in the absence of competition, they're both able to live in both environments pretty equally. And it isn't until you add competitors that now one species is able to persist in the one environment and not in the other environment. So there's that kind of environmental filtering that's caused by competition that wouldn't otherwise exist. So what's happening here? Competition is affecting the strength of filtering that's happening into one environment versus the other. But also the environment is affecting the strength of competition. So it's very much a reciprocal kind of interplay here. If we focus in on a particular species pair, these are two Licinia species that are both native to California. Same type of plot, but I'm just going to be focusing on them. Again, so this is a case where if we examine each of them in isolation, so not competing with each other, they're both way above in this persistence zone. They don't tend to really care if it's a dry or wet environment. Licinia california is slightly better in that environment than Licinia glabrata, but overall, the environment doesn't really seem to matter that much for them. They both do perfectly well. Again, if you then compete these two species together, there's two things that you can see. The first is that in a dry environment, they both fall above this persistence threshold. This is a case where you actually expect that these two species can coexist together. It isn't until you look in a wet environment that now this Californica species is actually driven below that persistence threshold, and you would expect glabrata to be the competitive dominant that excludes the other species. So you get coexistence in one environment and not in another environment. And again, it isn't until you actually cause these species to compete with each other that the wet or dry soil moisture environment actually affects their population growth rates at all, which is interesting. And so this is neat, but the environment can actually affect competition in two ways. And I'm going to tell you what those ways it can do that, and I'm going to then tell you which way it actually is. So again, let's say we have two species here. These are two grasses, just for cartoons, in one environment versus another environment. The first way that the environment can affect competition is by operating to affect their intrinsic growth rates, basically how well they do in the absence of competition just in a particular environment. Species might differ in which abiotic environment they like more. So here I've got this as little cartoons. Let's say in environment 1, the species does better. In environment 2, the species does better. Of course, we know that this is going to be the case. This is what a lot of us just intuitively think is happening when we're thinking about how the environment is affecting species in competition. But there is this other mechanism, and it's the mechanism that species actually hurt each other more or less when they're in different environments on a per capita basis. And so what I mean by that is here you can see these are per capita interaction strengths of one species onto the other. And what you can see here is that the environment might actually be enhancing some of those per capita interaction strengths, the degree to which this species hurts this species in one environment, but overturning that in another environment. And that's the one in particular that we don't have a lot of evidence in the literature of how strongly are these per capita interaction coefficients affected by the environment. And so I'm going to show you, this is kind of a runaround way of getting at this, but I'll show you that the per capita interaction strengths are in fact shaped by the abiotic environment and how we know that. So basically, you don't need to know too much about what these axes means. The x-axis is basically an estimate of niche differentiation between two species, and fitness differences are more just like overall competitive dominance of one species over the other. But importantly, it's only this x-axis where it's only the per capita interaction coefficients that affect the magnitude of these relationships, okay? And so that's important because if these per capita interaction coefficients are not modified by the abiotic environment, you would basically expect the environments to just be a vertical line because it's just affecting the intrinsic growth rates and it's not affecting those per capita interaction strengths. By contrast, if these aren't vertical lines, let's say it's purely horizontal or some kind of diagonal or something, this gives us evidence that the per capita interaction coefficients are in fact modified by the abiotic environment. So if I show you the actual data, the first thing that you'll notice is that there are a ton of non-vertical lines. And in some cases, they're mostly horizontal with only a slight vertical bent to it. And so this is telling us that the abiotic environment is very much so, in some cases very strongly, altering those per capita interaction strengths. And also, if you kind of average this across all species pairs, it's not like they're all doing it in the same direction. It's not like a wet environment is more competitive than a dry environment. It's more of this idiosyncratic effect where some species are being positively affected by that environment and some negatively affected by it. And overall, as you average between them, the environments just have the same average kind of effect on those per capita interaction strengths. And so this leads to this question that was posed in a 2017 Tree article of should environmental filtering be abandoned? And a response based on our data, I would say that first, yes, most species contain competitors and competitors strongly modify persistence. So it kind of makes sense that, you know, in what environment are you really going to see pure environmental filtering? But on the other hand, no, maybe we're just asking the wrong question, because I think what this actually does is reveals this really interesting interplay between species realized and fundamental niches. And what I will say too, I've actually, you know, I teach a really large second year ecology class, and I don't use that schematic anymore. I kind of more focus it on, you know, all processes being equal and how do they interact with each other and what are the outcomes. So take home here is that the processes do not operate hierarchically, but rather they're these reciprocal processes that all affect each other and interact. Okay, so that brings me to the next project that I want to tell you about, which is more about the spatial scaling of ecological processes. So again, we're kind of disregarding this hierarchical idea of species being filtered from some region down into a local community. But here we're really focusing on these different processes. We have dispersal or abiotic factors or the environment. And what I haven't argued in the previous section is that scale doesn't matter, because scale does matter. It just doesn't, it doesn't matter in this overly simplistic hierarchical way that we've thought about it. But I will say scale is something that's important to all of us. It's important to everybody probably in this room, whether you're thinking about it explicitly or implicitly. And the idea is that different processes do operate at different scales. It's not necessarily dispersal and abiotic and species interactions, but each of them could each have their own scale or series of scales at which they become important and at which they interact. This, of course, shapes the ecological patterns that we see in nature, whether or not be species richness or persistence or these other things. And it's also something that we're just assuming when we're kind of putting a box on a particular spatial extent in an area and we say this is the region and this is the local environment, which we often have to do when we do experiments because we need some way of knowing what our sampling block is and how big our experiment is, right? So if we're doing that, we're implicitly making this assumption about what scales a particular process might be important, whether or not it's dispersal or whether or not it's even adaptation or local adaptation in a particular area. This scaling isn't necessarily expected to be linear. And in fact, we think that it's probably nonlinear. And so I want to tell you about what John Wiens referred to as domains of scale, or Simon Levin as characteristic scales. And it's the idea that if we view scale as a continuum, as opposed to just saying small or large or medium scales, here's a continuum of very small at the left side, all the way up to very large as a continuum. And what these domains are basically extents of scale that are dominated by a particular combination of processes leading to a specific pattern. And what's interesting is you see there are multiple domains. So as you change the scale, you actually end up in these different regions where different processes tend to dominate. And what's interesting, if we just kind of arbitrarily pick some scales to conduct our experiments at, we might pick, okay, here's our local scale, here's our regional scale, but actually we've accidentally sampled in the exact same domain of scale. And so we wouldn't expect to see anything different happening among these two scales. Or we might pick these as our two scales, local and regional. We've actually missed a very important domain of scale in the middle that we haven't sampled. Or even more weirdly, we might happen to pick two scales that fall in this weird transition zone, which, you know, it would just be a confusion of processes. We wouldn't quite be able to know what's happening there. And the important part here is that we don't actually yet have experimental evidence of where these transitions occur in nature, despite this being such an important fundamental kind of question. And so my question here, I'm going to be focusing on dispersal and dispersal limitation in particular, and how this varies with spatial scale. The system that I work in, again, it's annual plants, but specifically those that occur in patches of serpentine soil in McLaughlin Natural Reserve in California. If you look at this landscape, you'll notice two things. You'll notice these areas that are this kind of gray, blue color, intermixed within this kind of matrix of greener stuff. And what this is basically these gray patches are this really harsh serpentine soil that plants in California have actually adapted to specialize on. And so they tend to be hyper diverse, tons of species. You can see this if I blow up a little picture of this community here. And they tend to be in these patches that are isolated from one another and separated by this matrix of exotic annual grasses. And so here you can see this is a patch, exact same patch that's shown in the Google image here. But all this yellow stuff is basically this monoculture of a Vina species that fill that matrix and make it very hard for species to just easily move from one serpentine patch to the other. And so what we'd want to do if we're trying to actually identify what these characteristic scales of dispersal are is we would want to experimentally isolate the spatial scaling of dispersal limitation. And so what do I mean by that? We can imagine that diversity in a landscape in each of these patches is basically the outcome of local environmental conditions. So just differences in the heavy metal content or nitrogen content, whatnot in these patches. And also the interaction with these regional dispersal histories. Have species been able to get everywhere in the landscape, or are they really just kind of stuck in a particular patch because they can't make it into other ones? If we actually wanted to isolate the magnitude of this dispersal limitation and do it at specific scales, we would actually want some way of experimentally removing and taking this dispersal history out of the equation, allowing species to get everywhere and seeing how diversity responds when it's purely responding to local environmental conditions. And what we would want to do actually is do this at multiple spatial scales in order to see at what point is dispersal limitation being most important. And I'm going to get at in a moment how I've actually tested this. But before that, I want to show you generally what you would expect if there's dispersal limitation. So here, we're going to be looking at species richness. And then also, this is the degree to which environmental gradients or conditions affect where different species are in the landscape. This first question that I want to answer is how strongly are species dispersal limited? So we could think about this 1 meter plot. These are species that are basically, exactly where they are naturally. Like I haven't done anything, I haven't manipulated dispersal, versus this 10-kilometer situation, which I'll describe in a minute, is basically what happens when you allow everything to get everywhere. So it's kind of two extremes: natural patterns, natural levels of dispersal limitation, everything being able to get everywhere. So if we compare those two extremes, if there's no dispersal limitation, basically it shouldn't matter if I allow everything to go everywhere. This wouldn't affect the number of species that are in the environment because everything is already where it should be, or it could actually cause a decrease possibly if I'm disturbing differences in abundance that happen naturally in the landscape of these species. However, if there is some dispersal limitation, you would see that this situation where I allow species to get everywhere should increase diversity. And it's really the difference between these 2 points that gives you an idea here that 15 species are dispersal limited if we see something like this. The second question is, how does this dispersal limitation impede species from sorting along environmental gradients? Again, if you give species access to a greater number of sites, they might be able to find a site that they are perfectly suitable in but had trouble accessing. And you would be able to see this in the response of the environmental explanatory power if you're comparing between the natural pattern and the one where you've completely removed dispersal limitation. And this is basically the space by environment interaction. It's how much does the environment affect or shape species distributions when you're removing that effective dispersal limitation. And then the last thing I can do is actually do this at multiple scales, like I've said, and ask this question of at what spatial scale does dispersal limitation manifest? Right here, I have the two extremes, but what happens in the middle if you remove things at 5 meters or 100 meters or 5 kilometers and so on? And so it turns out the best way to do this type of manipulation, which I consider quite clever, is to actually vacuum plant communities. And again, these are annual plants. So what that means is I can actually go out there with a vacuum when everything's senesced, everything's in the seed bank, vacuum it up and actually manipulate it in whatever way I want and put it back down and see what the consequences of that are. To actually remove dispersal limitation, basically what I do is I pool those seed banks at a variety of different spatial scales, either the 1 meter, which again is supposed to represent like the natural, like unmanipulated kind of diversity that you get, but mixing them at 5, 100, 5 kilometers, and 10 kilometers to sequentially allow them to get further and further and removing that dispersal limitation. And yes, surveying the community at the next growing season to see the consequences of this. So these plots, again, will look familiar. They're set up the exact same way as we just talked about. To answer this first question of how strongly are species dispersal limited, if I actually show you the data for this, what you can see is that basically in the natural ones that have only been moved about 1 meter, you get 12 species on average in those plots. If we compare that to the case where we've actually completely removed dispersal limitation at the scale of the whole reserve, you see this doubling in species richness. So the conclusion here is that maybe communities are actually half as diverse as they could be if we were to remove that dispersal limitation, which might be more similar to how communities looked historically prior to this invasion. How strongly does this dispersal limitation impede species from sorting along environmental gradients? Again, we can look at the other plot to see that. We all of a sudden see this jump from 35% of variation in occupancy patterns being explained by the environment up to about 52, 53, which, as you know, in ecology, that's a huge effect where sometimes we think 7% is a strong effect size. Here, there's this 20% addition of the explanatory power of the environment when you remove that dispersal history. And then the last question here about what spatial scale does dispersal limitation manifest? If we pop in all of the points here, what you can see is there's this really interesting S-shaped saturating curve, which is pretty cool. If I want to highlight the main things about this, basically there's this threshold below which dispersal histories don't matter. So it doesn't matter if you're moving things at 1 or 5 meters, those are all kind of close enough that like it doesn't really affect diversity in any way. But interestingly, the coolest part to me is what happens at these really large scales. If you manipulate things at the scale of half of the reserve, which is 5 kilometers, there's no difference between doing that and moving things across the entire reserve at 10 kilometers. And that's shocking to me that it doesn't have an effect because that's a huge difference in spatial extent. But we've kind of reached this saturation point of what exists in the species pool, which is pretty neat. And so I want to talk about why this might happen. It's this idea that spatial structure really underlies this characteristic scale of dispersal. I'm going to show this in 2 ways. The first is kind of what's happening at 5 meters. So here we have, this is just like a cartoon example of environmental conditions at a hypothetical site. We've manipulated these species pools at this scale. So that's what the little stars are. We've moved them a little bit. And what you can see is they're actually not experiencing a new environment. I've just moved them 5 meters, not a big effect. At 5 kilometers, I've actually allowed them to cross this inhospitable matrix. I've allowed them to experience a greater range of environments. And at that point, we've kind of sampled all of the environmental variation that exists. And it doesn't matter if you're at 5 or 10 kilometers, that's going to be the maximum response that you see. And this really overturns that notion that the dispersal and environment exists as a hierarchy of processes. Because if we didn't have this spatial patterning in environmental conditions, basically dispersal scales wouldn't necessarily matter, and you would expect these populations to kind of permeate the landscape over time. But it's because of this environmental structure that that doesn't happen. To show you this another way, I want to show you some work that I've been doing with some folks, Yuval, Matthew, David, and also a bunch of other people as part of a working group funded through the CNRS and QCBS. And here we're using a meta-community model to kind of simulate this interplay between the scale of the environment and the scale of dispersal, plus a few other things, because that basic relationship has been examined before. But what I want to show you, so this is a landscape here specifically dealing with an environment that has a very fine-grained spatial scale. So you can kind of see this. This is biomass distributions across that landscape. And the environmental scale here is 10. And we're looking at this at a range of different dispersal values, 1, 3.2, 10, 32, and 100. The first thing I want to point out here is that if we look at these first two blocks, they actually look the same. There's no difference between them that I can resolve with my eyes. If we look at these other three plots that are same landscape, but dispersal's really ramped up, we actually see that dispersal starts to matter and that pattern changes as we ramp up that dispersal scale. If we compare this to a landscape that has a coarser grain to it, so you can kind of see the pattern here, it's coarser grained. Specifically, the value is 32 in this case. We actually see that these first three panels now are the same, basically the same pattern. And it isn't until you get to these really high dispersal values that dispersal starts to matter at all. And the really interesting thing here that I want to point out is the transition at which this occurs is exactly the scale at which dispersal is at a smaller scale than the environment versus dispersal being a higher scale of the environment. And what this means to me is that this transition point between dispersal mattering and dispersal not mattering is entirely set by the scale of the environment or that environmental template. So again, this interplay between the environment and dispersal. And so I think the broader take-home message here, if we're thinking about scale in general, is that scale of one process, so let's say the environment in this case, can actually cause the emergence of characteristic scales at other processes. If the environment didn't have that particular spatial scale, if it was just completely fine-grained, dispersal wouldn't matter at all. And it's because of that environmental template that dispersal matters, which is interesting. Okay. So this project is really cool, but it's far from perfect. And there's a few things I want to do next. The first is test how persistent these outcomes are. We have repeated this study for 3 years and shown that it is persistent, but there's a lot of really cool things we can actually do at looking at new colonization events and extinction events. And I have a student, Jessica, who's working on this right now. And the second, we can actually ask how strongly dispersal is driven by the abiotic environment versus competition. And so I want to tell you really quickly about a project. I'm not going to show you data for this one, but I do want to just mention it because it's really cool. And this is work by Megan Soyka, who's going to introduce herself.
Hello, I'm Megan.
So Megan did her master's with me on this project and another one. And now she's actually moved on to do a PhD in Lauren Shoemaker's lab. And we're actually all collaborating on this project together, which is super fun. But it's on the spatial scale of species persistence. Where basically Megan replicated the experiment that I've just shown you, except what she's done additionally is able to remove competitors or leave them intact and actually perform an invisibility experiment where she's looking at like, okay, if we put these populations in this particular plot of this particular environment, are they going to be persistent or not? But again, doing this across the landscape in a whole bunch of different environments at a whole bunch of different scales. She's done this with 4 species. She has 3 nested scales from the plot to grid to site level. And she's just got like thousands of transplants across this 10-kilometer scale. Really impressive photo of what this looks like. This is just one of 18 of her sites to give you an idea of the scale of this project. And the projects that I've shown you already, the first one with the environment and competition is looking at this interplay and interaction between the biotic environment and the abiotic environment. The second one is looking at dispersal and how that interacts both with the biotic and abiotic environment, because those are both shaped by dispersal. But then this third project that Megan's working on right now is actually going to combine all of these 3 things together. We're going to be able to look at persistence, how it's affected by dispersal, the biotic environment and abiotic environment. And it's going to be doing this in a natural landscape where I'm actually not aware of another experiment that has done something like this that can look across multiple scales and look at persistence and the underlying drivers of those patterns. So I'm very excited about it. Okay. So that is part 1. Part 2 is going to be a bit shorter. So if you're worried about time, I think we're fine. Don't worry about it. And here we're going to be focusing more on the evolutionary ecology side of things. And one thing I want to point out is my lab is working on a number of themes right now. And so I wanted to throw those out there on the evolutionary side. We're working on character displacement, speciation, macroevolution, gene flow, community evolution, balancing, selection, all of these things. And all of them, we're examining them, again, through this perspective of updated ecological theory and linking population demography to persistence and coexistence. But I'm not going to be talking about those projects today because I really wanted to focus on something that explicitly had to do with the theme of the talk, which is scale. But I do want to plug that evolution is happening right now. And I have 2 students who gave talks, which are all recorded. You can go check them out. They're really good talks, so do check them out. The first is by Mia Waters. She's looking at history's influence on population divergence via ecological persistence. And Jawad Sakarchi, who's working on the evolution of competitive ability. So yes, keep their names in mind if you're registered for evolution. What I actually wanted to talk to you about was something that arose via something that I noticed when I was in the field a lot. And so again, I did this big vacuuming experiment. I went back 3 years in a row, and there's something that I noticed. And what I noticed is that what this is, these are 2 plots. They're in different parts of the landscape. One's really harsh, the one on the left, and one's really benign. And this is what it looks like in a really good climate year. The thing that's obvious, I think, is that they look very different from one another, right? One's very lush and productive, and the other one isn't. If we compare this to a harsh year, this is a bad climate year, really droughty. It actually looks like they look pretty similar, which is interesting. And if I want to look at this a slightly different way, this is the same plot in 2 years during a good year and a bad year. Not a big effect, right? It's still unproductive, still doesn't look great. But if we look at that productive site in a good year versus a bad year, there's actually a really huge effect where it's very much hammered by this bad climate. And what does this have to do with scale? What I think this is this very interesting interaction between spatial landscape heterogeneity and temporal climate variability. Another way to say this is that temporal variation is actually not being realized equally across space. So a little cartoon of that, if we compare site productivity over time of those plots that are in an unproductive soil versus a productive soil, basically what you can see is there's a lot more variation in that productive soil than in the unproductive soil. Now we're thinking very generally what this concept is that I'm describing. It's actually something that we refer to as a cross-scale interaction. It's basically, in this case, the exact same process, which is the environment, is operating and interacting actually with that same process at a different spatial scale. So the large scale climate environment, small scale soil environment, and having this interesting interactive effect. But also more specifically, it's an idea called co-limitation. So when the environment, the soil environment is very harsh, it doesn't really matter how good the climate is. You can't really respond as strongly as if you would if you were in a productive soil, because there's kind of 2 things that are limiting you. And so here how I'm examining this, I think it's something that's very relevant for spatial coexistence mechanisms. But here in particular, I'm going to be talking about the evolution of bet hedging strategies specifically. And this is, the question here is, how does bet hedging evolve along a productivity or variability gradient? And this is work by Kelly Sliman.
Hi, I'm Kelly.
Who did her undergraduate thesis with me and is starting a PhD in the Agri lab at Cornell. And the basic idea here is that we're looking at sites that vary in their productivity. We have sites that are less variable and sites that are more variable. And of course, we know that with bet hedging theory, we would expect that something like dormancy, which is supposed to be a mechanism for coping with temporal variability, you'd expect it to have this positive relationship where it actually evolves to be more strong in those variable environments and less strong in those less variable environments. So to test this, we did a germination assay with 23 populations collected from those sites that span that productivity gradient, some sites that are really unproductive and some sites that are very productive. We grew them in a greenhouse for 1 generation and then did a germination assay to actually measure the degree of dormancy. And when we do this, so again, to remind you what the prediction is, this positive relationship, we actually find the opposite. And this is very significant, opposite negative relationship here.
How surprising.
It is very surprising. We're very much scratching our heads about what happened here. This made literally 0 sense to us. Why would a strategy that's great for coping with environmental variability actually be higher in an environment that's less variable? So I was thinking about this a lot. And then one day it hit me. And the question here is, what if dormancy actually evolves to combat fine-scale spatial heterogeneity? So again, these are our plots. We have our unproductive plots and our productive plots. And what you can see is that basically, there's just way less plant cover in the top one. It's rockier. You can imagine that there's fewer microsites that are actually suitable for germination. So maybe dormancy is actually a mechanism for coping with this spatial heterogeneity rather than time. And so to test this, I did want to test this. We went back to sites that had lower high productivity. And I wanted to test the proportion of those plots that were actually suitable or unsuitable to germination. And the prediction here is that unproductive sites have a higher proportion of microsites that are unsuitable to germination. So that's what this would look like. Yeah, more unsuitable sites in the low productive sites than the high productive sites. And we test this, basically it's kind of basic in this case, but stabbing it with a nail and saying like, can this be penetrated at all? So very much a conservative measure, like how rocky is this, that kind of thing. And when we do that, that is in fact what we find. Those unproductive sites, just if you were a seed and you were to land in 80% of that site, you would be unable to actually root there because you're probably encountering a rock. So this is supported. But it was making me think more in general about this idea about the collapse of space and time. And part of this was stimulated by watching the movie Interstellar, which is all about black holes and how time and space can become one. And specifically that love and also dormant life stages are things that may be able to actually transcend time and space. And so what do I mean by that? So if we're thinking about this plant that has these different sites that it could access, and we have conditions that are variable through time, this plant produces a seed, that seed is dormant. The first environment that seed experiences is this blue environment. The environment changes, it doesn't move anywhere, it's staying there. It's experiencing now a different condition, changes again, and it's experiencing a different condition. We can compare this to what happens with variability in space. If that seed can actually move around, at time one, yes, it's still experiencing that blue environment. If it moves a few centimeters over, it might actually be sampling a very different environment. And again, moving a little bit further, it's experiencing a different environment. And so what you can see is that different mechanism here. This is temporal variability on one side and spatial variability on the other. But from the seed's perspective, they're kind of experiencing it in the same way. And to me, this is entirely plausible because this can happen over a scale of 10 centimeters in these unproductive sites where the microsites are suitable adjacent to unsuitable microsites. So maybe variability in space and time are actually experienced in the same way from the perspective of these dormant individuals. And really this microgeographic variability may cause time and space to be experienced as one axis, or kind of collapsing them. And so one thing I want to tell you about, which is a little bit of an aside, this is the last thing I'm going to tell you about, it's an aside, it's not even directly relevant for this project, but it's something that has emerged in all of the work that we've been doing, especially when we've been conducting invisibility experiments in the field. And it's the idea that, I'll introduce this first, we have mean individual fitness on the left and per capita population growth rates on the right. And if you're an evolutionary biologist, you might think about this more, mean individual fitness is the realm of evolutionary biology. If you're an ecologist, you might think more on this side, the realm of population ecology. And of course, the interlinks between these two things, we've heard many people say this, this is really what binds ecology and evolution, right? That mean individual fitness can affect population growth and population demography can affect fitness. And so that actually causes eco-evolutionary dynamics. But one thing that it's even more fundamental than that, but maybe not something we think about all the time, is that these are actually the exact same quantity. So if we're thinking about mean individual fitness, that is actually identical to per capita population growth rate. So I just want to show you something really quickly that illustrates why that's kind of interesting in a way that you wouldn't expect. So here, I'm going to be scaling populations to individuals.
Hey, I'm Mia.
So Mia is a PhD student who's been doing invisibility trials with a bunch of different populations in the field. And actually, what's not important here is the competitor without competitor thing. I'm going to unpack this for you, and then you'll see what's interesting here. But we have individual fitness on the y-axis, and these are all just different populations. They happen to be in a competition, no competition environment, but that's besides the point here. Each dot is a separate population average, and that average is composed of 10 individuals in the population. One thing that I never really thought about is how there might be this disconnect between what's happening at the individual level and what's happening at the population level. And so we might think that, if a population has an average per capita growth rate of 20, that means that all of these individuals are producing 20 seeds. It's kind of like evenly distributed among individuals. And so if we were to take those 10 individuals, let's say this is a line that's following 10 individuals in a population, we might think if this is the population average, that all of them are kind of contributing equally, right? And these would be flat lines. By contrast, if we actually look at what's really happening, these are kind of like rank abundance plots, but they're actually individuals and versus individual fitness. What we can see here is that some individuals are actually leading these populations. So if we look at this one population, which has a very high population growth rate, it's actually driven by a single individual that was very high performing, and then a bunch of crappy ones, and in fact, a lot of zeros. And we can compare that to a population here that all individuals did okay, but none of them super great. And so what I actually think is there's this really interesting thing. We've seen this in all of our data sets at this point. The populations are really being led by a few exceptional individuals. And I keep trying to think about what that means for ecology and evolution. If we're trying to say like, how adapted is this population? This one has high mean fitness, this one has low mean fitness. What does it mean if individuals are all kind of contributing in a high performing way versus some of them just leading those populations completely forward? And what does that mean ecologically for the distribution of individuals in a landscape if, really it's this one individual over here that produced a ton of stuff and everybody else failed, and leading to these kind of clumpy distributions of abundance in a landscape? And so that's just a little hint at some of the things that we're thinking about doing right now. But the take-home messages from this talk that I want to reiterate about scale is that processes do not operate hierarchically, but rather reciprocally, and they affect each other reciprocally. I've told you that the scale of one process can cause the emergence of characteristic scales of other processes, which is a very important kind of key, interesting point that, very neat. The microgeographic variability may cause time and space to be experienced as one axis, and that populations may actually be led by a few exceptional individuals is something we see over and over again. And this really warrants more of a look at the scale of individuals. And so really what I hope to do is combine all of this work together, specifically the evolution and the scale of ecological processes, where I've told you that scale affects those ecological processes, which then affects those patterns. There's been some previous work that looked at the link between scale and adaptation. And really what I want to do is close all of these links here, is link scale to the processes, the ecological patterns, and how that feeds back to affect adaptation and eco-evolutionary dynamics. Specifically taking this approach that I've been using with population ecology and community ecology, and really putting in more of this finer scale look of what individuals are actually doing, and what does that mean ecologically and evolutionarily. And so with that, I will thank all of these people and also the organizers of this session for inviting me here, and all of you for listening. Thank you.
Okay.